Revolutionizing mental health. New discoveries on the 5-HT1A brain receptor. Revolutionary brain discovery could transform how we treat mental illness. New York, New York, August 1, 2025. The battle against depression, anxiety, and psychosis may soon enter a new era, thanks to groundbreaking research that has uncovered critical secrets about how our brains process emotions at the molecular level. Scientists at the Icahn School of Medicine at Mount Sinai have just pulled back the curtain on a mysterious brain receptor that could completely revolutionize mental health treatment as we know it. The groundbreaking study, which appeared online in the August 1st edition of Science Advances, https://www.science.org/journal/CIADV, zeroes in on the 5-HT1A serotonin receptor, a crucial component in our brain's mood regulation system and a primary target for both conventional antidepressants and emerging treatments like psychedelic therapies. Despite its enormous importance in mental health care, this receptor has remained something of a black box to scientists until this remarkable breakthrough. Think of this receptor as the brain's emotional thermostat. It constantly adjusts how neurons respond to serotonin, which is absolutely fundamental to our emotional well-being, thought processes, and overall mental state, explains lead researcher Daniel Wacker, PhD, https://profiles.mountsinai.org/Daniel Wacker, who serves as assistant professor of pharmacological sciences and neuroscience at Mount Sinai's Icahn School of Medicine. Our research essentially decodes the instruction manual for this thermostat, revealing which buttons do what, how it calibrates neural signals, and where its capabilities end. This deeper knowledge opens exciting possibilities for creating your targeted treatments for conditions ranging from depression and anxiety to schizophrenia. Through cutting-edge laboratory methods, the research team made the fascinating discovery that the 5-HT1A receptor is naturally predisposed to prioritize specific cellular communication pathways over others, regardless of which medication is targeting it. Nevertheless, different drugs can still modulate the intensity with which these pathways activate. In one notable example, they found that the antipsychotic medication asenapine, marketed as Safras, uniquely engages a particular signaling channel due to its relatively mild interaction with the receptor. To delve deeper into these intricate mechanisms, the scientists employed a two-pronged approach. They conducted experiments on laboratory-cultured cells while simultaneously utilizing state-of-the-art cryo-electron microscopy, a revolutionary imaging technique that visualizes molecular structures down to nearly atomic detail. Their investigation concentrated specifically on how various medications activate the 5-HT1A receptor and how this receptor interfaces with internal cellular messengers known as G proteins. But here's where it gets fascinating. The different signaling pathways controlled by the 5 new insights into serotonin 5-HT1A receptor signaling toward next-gen mental health drugs. Unlocking the secrets of serotonin receptor signaling could revolutionize the way we approach mental health medications. Researchers at the Icahn School of Medicine at Mount Sinai have made significant strides in understanding the intricate workings of a vital brain receptor, which may lead to the creation of advanced antidepressants and antipsychotic drugs, published in the online edition of Science Advances. This groundbreaking study centers on the 5-HT1A serotonin receptor, a crucial component in mood regulation and a frequent target for both conventional antidepressants and innovative treatments like psychedelics. The research paper is titled Structural Determinants of G Protein Subtype Selectivity at the Serotonin Receptor 5-HT1A. Despite its importance in clinical settings, the 5-HT1A receptor has remained largely enigmatic, with many of its molecular and pharmacological characteristics not thoroughly explored until now. Think of this receptor as a control panel that regulates how brain cells respond to serotonin, a vital neurotransmitter that influences mood, emotions, and cognitive functions, explains Dr. Daniel Wacker, the senior author, and an assistant professor of pharmacological sciences and neuroscience at the Icahn School of Medicine. Our research illuminates the workings of this control panel, what mechanisms it activates, how it modulates signals, and its operational boundaries. This enhanced understanding could pave the way for improved treatments for mental health disorders such as depression, anxiety, and schizophrenia. Utilizing cutting-edge laboratory techniques, the research team found that the 5-HT1A receptor is inherently predisposed to favor certain cellular signaling pathways over others, independent of the specific drug targeting it. Nonetheless, medications can still modulate the intensity with which these pathways are activated. For instance, the antipsychotic acinapine, marketed as Safras, was shown to selectively engage a particular signaling pathway due to its relatively mild activity at the receptor. To delve deeper into these mechanisms, the researchers employed a combination of experiments using lab-cultured cells and high-resolution cryo-electron microscopy, a state-of-the-art imaging technique that reveals molecular structures with near-atomic clarity. Their investigation focused on how various drugs activate the 5-HT1A receptor and its interactions with internal signaling proteins known as G proteins. The different signaling pathways governed by the 5-HT1A receptor are associated with various aspects of mood, perception, and even pain. As scientists gain a clearer understanding of which pathways are activated and the mechanisms behind them, they can design medications that target specific symptoms or conditions while minimizing unwanted side effects. Our research provides a molecular blueprint of how different drugs interact with this receptor, activating or inhibiting specific pathways that affect brain function. States Dr. Audrey L. Warren, the first author of the study, and a former student in Dr. Wacker's lab, now a postdoctoral researcher at Columbia University. By comprehending precisely how these drugs engage with the receptor, we can begin to forecast which strategies may yield more effective or targeted treatments and which ones are less likely to succeed. This represents a significant step toward crafting next-generation therapies that are more precise and have fewer adverse effects. In a particularly unexpected finding, the researchers identified that a phospholipid, a type of fat molecule present in cell membranes, plays a crucial role in directing the receptor's activity, functioning almost like a hidden co-pilot. This is the first instance of such a role being documented among the over 700 known receptors of this kind in the human body. While traditional antidepressants often require weeks to show effects, scientists are optimistic that this newfound understanding of 5-HT1A signaling could clarify these delays and lead to quicker-acting alternatives. This receptor may provide insights into why conventional antidepressants take so long to work, Dr. Wacker notes. By grasping its molecular functioning, we can chart a clearer course toward developing faster, more effective treatments, not only for depression, but also for conditions like psychosis and chronic pain. It's a crucial piece of the puzzle. Looking ahead, the research team intends to further investigate the role of the phospholipid cofactor and assess how their laboratory findings hold up in more complex experimental settings. They are also working on translating these discoveries into practical compounds that could evolve into future psychiatric medications, building on their previous successes with drug candidates derived from psychedelics. Revolutionary brain receptor breakthrough. Could this cure mental health disorders? What if we told you that the key to faster, smarter mental health treatments might lie hidden in the tiniest structures of your brain? A team of scientists at the Icahn School of Medicine at Mount Sinai has just cracked open a biological puzzle that could redefine how we treat depression, anxiety, schizophrenia, and even chronic pain. Their findings, published in Science Advances, reveal never-before-seen details about the 5-HT1A serotonin receptor, a molecular switchboard in your brain that controls everything from mood to perception. But here's where it gets controversial. This research challenges long-held assumptions about how drugs interact with this receptor, sparking debates about the future of psychiatric medicine. For decades, the 5-HT1A receptor has been a prime target for antidepressants and emerging psychedelic therapies. Yet, despite its importance, scientists struggle to fully grasp how it chooses which signals to send inside neurons. The breakthrough? Using cryo-electron microscopy, essentially a superpowered microscope, they captured atomic-level snapshots of the receptor in action. These images show how the receptor selectively partners with specific G proteins, molecular messengers inside cells, essentially picking which pathways to activate. This inherent signaling bias, as the researchers call it, means the receptor doesn't just react to drugs. It has its own built-in preferences. And this is the part most people miss. Even powerful medications can't override these preferences. For example, the antipsychotic asenapine works by subtly nudging the receptor toward one signaling route over another, which directly impacts both effectiveness and side effects. Now, imagine a lipid molecule, a type of fat, secretly acting as a co-pilot for this receptor. That's exactly what the team discovered, a phospholipid embedded in a cell membrane that fine-tunes receptor activity. This finding upends previous models of receptor function, suggesting that the very fabric of your brain's cell membranes plays an active role in mental health. Think about it. Could this mean that future treatments might target not just proteins, but the entire cellular environment? The implications are staggering. Current antidepressants often take weeks to work because they're fighting an uphill battle against these natural signaling delays. By mapping the receptor's precise control panel, scientists could design drugs that bypass these delays entirely, delivering relief in days rather than weeks. The study's lead researcher, Daniel Wacker, PhD, compares the 5-HT1A receptor to a sophisticated airplane cockpit. We're finally seeing the buttons and levers that make this system work. This knowledge is the blueprint for creating next-gen therapies with pinpoint accuracy. Audrey L. Warren, Ph.D., who spearheaded the research during her time at Mount Sinai, adds that this work opens a new language for drug development. Instead of grouping medications into broad categories, we might soon classify them by their exact molecular actions, like how a car mechanic would analyze each component of an engine. This level of precision could drastically reduce side effects like drowsiness or weight gain, which plague many current treatments. But what about the skeptics? Could this lipid-based model oversimplify brain function? Some experts argue that focusing too narrowly on individual receptors might miss the bigger picture of how the brain's networks interact. We want to hear your thoughts. Do you think targeting these molecular buttons will truly revolutionize mental health care, or is the brain too complex for such precision? Share your perspective in the comments below. Looking ahead, the team plans to test how manipulating this lipid-receptor relationship affects real-world behavior and drug responses. They're also working to translate these insights into actual medications, building on recent successes with psychedelic-derived compounds. This study isn't just a scientific milestone. It's a bridge between cutting-edge structural biology and practical psychiatry, showing how advanced imaging techniques can solve age-old mysteries about the brain. For the millions struggling with mental health disorders, these discoveries offer more than hope. They provide a clear roadmap toward treatments that work faster, smarter, and with fewer drawbacks. As Dr. Wacker puts it, we're not just studying proteins anymore. We're learning to speak the language of the brain itself. Subject of research: cells. Article title: Structural determinants of G protein subtype selectivity at the serotonin receptor. 5-HT1A news publication date: August 1st, 2025. Web references: Science Advances. Journal references: Warren A.L., Zilberg G., Abbasi A., Abraham A., Yang S. Structural determinants of G protein subtype selectivity at the serotonin receptor. 5-HT1A. Science Advances, 2025. Image credits from A.L. Warren et al. Structural determinants of G protein subtype selectivity at the serotonin receptor. 5-HT1A. Science Advances, 2025. Licensed under CC by NC 4.0. Keywords: mental health. Tags: 5-HT1A serotonin receptor research, anxiety and depression therapies, breakthrough mental health treatments, chronic pain treatment innovations, cryo-electron microscopy applications, Icahn School of Medicine research findings, molecular insights into brain receptors, next-generation antidepressants, psychiatric medicine advancements, schizophrenia treatment breakthroughs, serotonin signaling pathways, targeted drug development for mental health.